CRISP-ID is a novel application for the exact determination of indels resulting from next-generation gene editing technologies. Whilst these tools, especially CRISPR, have been widely adopted, the precise characterization of indels in resultant clones remains challenging. The wild-type CRISPR-Cas9 complex is targeted by a guide RNA to a specific region of the genome, where it introduces a double-strand break. The cell's endogenous DNA repair mechanisms repair this break by non-homologous end joining. However, this is prone to errors incurred in a largely random manner. In a diploid cell, this generally results in two different alleles. Sanger sequencing of a mixture of two different alleles results in a mixed spectrum. This problem is classically solved by cloning the mixture of alleles into a bacterial vector and sequencing several colonies, thereby sequencing each allele individually. This is, however, laborious and costly, especially in high throughput. CRISP-ID directly analyzes the Sanger sequence of the mixture of alleles. CRISP-ID can resolve a mixture of up to 3 alleles, making cloning for indel characterization unnecessary for diploid and triploid cells. To get started, you will need a Sanger sequencing chromatogram file in an AP1 or SCF format. You will also need a reference sequence for your targeted region. You start by uploading your Sanger sequence chromatogram file and wait for the upload to complete, where you will be met with a graphical representation of your mixed sequence. Next, paste in your reference sequence for your targeted region and hit Analyze. You will be met with an alignment of your reference sequence to your alleles. The alleles are also presented in FASTA format. Here we have a file where a problem has occurred. The application has issued a warning that no alignment with the reference sequence could be found. Upon inspection of the chromatogram, we can locate the problem. At this location, there's an unusually high background that has been picked up as signal by the base calling algorithm. With the orange marker, the software has indicated that this is considered to be the start of the spectral overlap. The real start, however, is located several hundreds of bases further downstream. We can solve this problem by increasing the threshold for the background cutoff, but in this particular case, the background problem only occurs in a single location, and we can easily bypass it by trimming the first 80 bases instead of the default 50. Now the marker has been placed in the correct location. Next, rerun the analysis to obtain the correct output. If you have any further queries or suggestions, please contact us using the contact form.